Am I supposed to start?
Ah, ya lo inicié.
Okay,
It's
when
messed up now.
start to share the, okay.
Okay, go ahead and start, yes?
No,
See?
not yet, not yet.
No?
Yeah, we can see, but not yet. You can stop sharing your screen.
Oh, sure.
Okay, Guillermo, when you say that I can start, I can start.
Besa, Alberto, que ya está transmitiendo.
Okay.
Ya está, Alberto, ya está.
Muy bueno. Bien. Muy buenos días para todos, muy buenas tardes para los que estamos en el hemisferio sur. Bienvenidos a la segunda parte del ciclo de seminarios de actualización de métodos moleculares aplicados al diagnóstico y control de HLB de los cítricos. Este es un ciclo de seminarios, webinars, para los participantes del componente 1 del proyecto PROCISUR, Desarrollo y Promoción de Herramientas Innovadoras para la Prevención y Mitigación del Efecto del HLB en los países miembros del PROCISUR. En este martes 24 de agosto de 2021 tendremos una serie de seminarios, van a ser 2 seminarios, webinars, sobre utilización de ARN de interferencia para el control de insectos. Los seminarios pueden ser en portugués o en inglés, así que tendremos los seminarios en otro idioma, no en español. Para la gente que luego nos siga en YouTube, cuando esto quede grabado, podrán ver los subtítulos abajo. Los seminarios estarán a cargo del responsable del componente 1, que es el doctor Eduardo Andrade, el cual hará la introducción de los speakers y de los oradores, y luego llevaremos las preguntas al final. Para los que nos están siguiendo desde YouTube, pueden hacer sus preguntas en el canal de YouTube, las vamos a ir copiando, y luego de eso tendremos en la sesión de preguntas y respuestas. So, Eduardo, the channel is yours.
Obrigado, Alberto. We are going to make this all this seminar in English because we have a guest, an invited guest from the United States, so everybody can catch up all the information. We're going to make this all the seminar in English. We have today 2 speakers. The first one is Professor Moisés Zotti from Universidade Federal de Pelotas, Rio Grande do Sul. He's going to talk about RNAi in insects from bench to field. So Moisés Zotti is an agronomic engineer with PhD in plant pathology. He's been working on entomology, molecular biology, bioinformatics, and insect pest management. He's an excellent collaboration with the group from Ghent University. He works on RNAi, some related topics. So the second speaker is Rene Hunter from Horticultural Research Laboratory, United States, from USDA in Florida. Wayne is a plant entomologist. He's a long work in insect biology, insect molecular biology, and mainly in psyllids and other insect vectors. He was the first one who made the first draft genome from psyllids, so he's a well-known person. And he's going to talk about RNAi to control Asian citrus psyllid. Okay, so I'm going to invite the first speaker, Moisés Zotti, so he can share his screen and start his presentation, please.
Okay, Eduardo, can just confirm. Can you hear me? Okay. Okay. No. Yes. Okay. So thank you, Eduardo, for inviting me to come here to present a little bit on our research on RNAi. So the proposed subject for today is RNA in insects from bench to field applications. So, and the subject on RNAi, I feel, very comfortable to talk because it's a subject you are involved in a lab on the last 5 years. And I will just switch off my camera here and then, because I'm afraid on the connection, so we'll stop my, okay, now it's fine. So it's RNAi is a subject you are being involved in a lab on the last 7 years. I was promoted already of 2 PhD students, and now I have more 2 PhD students in my lab. They are working with RNAi. Normally, the first slide in every presentation, I just pass one overview in some subjects you are involved in the lab. So we focus on sustainable pest control. So we do research, and also we have, as all university professors, the function of human resource formation. So we use insects for do our research, but also we teach students in molecular entomology. So we have projects with hormones and receptors in insects, like ecdysone receptor. We like very much to work with this receptor. Some work with GPCRs. We do a lot of cell-based screening, especially for ECR ligands. We do sequencing of transcriptomes. We do RNA-seq, of course, RNA interferencing. We have some projects with ants and termites, and which we look for new antibiotics because these organisms, they have association with bacteria that produce a lot of antibiotics. And pollinator, of course, and we like very much to work in between wet lab and dry lab. So we like very much to work with molecular modeling, protein dynamics, and also use a computer to find new molecules. So this is one overview of the main subject we develop in the lab. So what I have prepared for this presentation, I divide in 6 main parts. What is RNA interferencing, RNA mechanism, how it works, possible applications, one brief experiment design, and transgenics versus non-transgenic methods, like a topical applied RNAi, and some perspectives. So what is RNA interferencing? Well, I think that most of people here, they know already what is RNAi, because we have been using for more than 3 decades maybe. Is a post-transcriptional gene silencing. So you have here DNA, and then DNA, because of the central dogma of biology, it's converted to mRNA, and then it's translated into proteins. So what RNAi does is mRNA degradation. So, and then you don't have protein formation. Very straightforward. And if this protein is related to, say, a vital function, and this protein is not formed, probably the insect will die, or at least reduce its performance. So, and today RNAi is considered like a toolbox, because if you are able to silence the genes, and then you can use it for pest control, like kill insects without harm beneficials, you can use to silence the genes and pathogens. Also, you can use it to protect insects, like to protect beneficials, like honeybees from varroa, nosema, and viruses. Of course, for crop improvements, RNAi has been used for a long time for crop improvement. and also as a resistance suppressors. Like if you have a resistant insect or a weed, which is producing a high amount of one enzyme, and then you can use RNAi to knock down that enzyme. And then in combination with the pesticide, the regular pesticide, you can maybe postpone. This is very important because we know that in Brazil, especially, I mean, a lot of BT crops, they are being not used anymore because the insect become resistant. And then if you have this technology, you can knock down the enzyme responsible for insect resistance, and then we can maybe postpone the technology for a couple of years in the field. This is very important. So why RNA interference has a unique mode of action? Well, I do some comparison here. If you have an agent of pest control like a predator, and then the target will be the whole organism, and then you have one physical elimination of pests. If you have a biopesticide like a BT toxin, and then you act at organ or cell level, and then you have cell disruption or organ function. If you have a chemical pesticide, you act on protein level, and then you have a disruption of protein function. If you have a radiation, you act on DNA level, and then you have a DNA damage. And then you have RNAi, you act on RNA level, and then you have degradation of a specific mRNA. As you can see here, RNAi acts on RNA, and comparing to all above is unique. So that's why RNA interference has a unique mode of action. Also important to do some comparison here. If you do like the traditional improvement to get a plant resistant to an insect or a fungi, probably will change a lot of genes, probably more than 10,000. And mutagenesis is complicated to say how many genes will change. Transgenics, 1 to 4, this number of genes normally you modify. And when you work with RNAi, probably will affect 1 or 2 genes, the same as CRISPR-Cas9. But how RNA is in action? Here I have one very straightforward example that you can see. Normally people, when they start to work with RNAi in the lab, they use this as a proof of concept. I mean, people like because you can see the phenotype very clearly. So lacase 2 is one enzyme responsible for cuticle tanning. And when you knock down that enzyme, you see the insect looks whitish. And as I told before, people normally they use this because you can see very clear. I mean, if you inject this and you see this color in the insect, okay, RNAi is working. And just a little bit more details here, how scRNA, which is one of the pathways explored for pest control works. So normally double-strand RNA comes into the cell, and then this double-strand RNA is sliced by one enzyme called dicer in small RNAs, and then this small RNA is coupled with one enzyme called argonode to form one complex, RISC. RISC means RNA induces silencing complex. And then this complex find target RNAs, which is complementary to that sequence, and then argonode has one slice motif, which will slice the mRNA, so you don't have protein information. So how is this technology RNAi comparing to other, regular pesticides? So we know already that regular pesticides, they have a quick resistance development. In many cases, 2 to 4 years. They are not specific to target insects. They have broad spectrum, and they kill many beneficiaries like bees, pollinators, and of course, without pollinators and without natural enemies, insects that they are not pests, they become pests. So RNAi has a less chance for resistance development, of course, based on the current mechanisms that you know. And it's more specific to target and therefore not affect beneficiaries. I mean, RNAi, it's like it's tailor-made. You can customize your sequence to target specific genes and affect only that specific genes in that insect you want to control or weed. I mean, this image I illustrate here. When you design a pesticide, a regular pesticide, a molecule to act in a specific protein, I mean, you see here the binding pocket. Normally, the binding pocket of proteins, they're larger molecules. I mean, it's almost impossible to define here how many contact points one pesticide will interact with this protein, because there are probably 100, maybe 1,000 of possibilities. But when you design a strategy based on RNAi, all information you need is in the sequence. What you have to do is only synthesize a double-strand RNA complementary to that region of the gene you want to silence. So it's I mean, at least it's more easy than you design a regular pesticide. And what were the possible applications for RNAi? Well, we can use for functional genomics, like you do some gene knockdown and then you can discover the gene function. You can use some therapeutic applications like silence viruses or disassociate genes, which is already in the market. And of course, for crop protection, you can target a pest using RNAi technology. This is some illustrations from, is the same as I presented before. You can use for functional genomics, and when you silence lacase 2, you can see the insects when they have this gene silence. I mean, the insect become whitish. And how you can use it so for crop protection? Well, you have to select critical genes in the pest organisms for knockdown. Pest organisms can be insects, can be weeds, or can be pathogen or plant pathogen. So, and then you have a lethal or a sublethal effect. Well, this, if you want to control insects, sometimes you not need the effect to be lethal. Maybe sublethal effect, the insect will reduce the performance, and then by the end of the process, will be the same. And how you can do this? Well, you have to administrate to provide double-strand RNA to the target pest. You can do this through transgenic plants. We know very well that transgenic plants, they work very well. How many years you are using BT crops? For many years, they work very well. But you can also spray double-strand RNA. You can do trunk injection or in drench. And we'll talk later. This is very important. I mean, how you deliver the technology to the field. So this is a seminal article published in 2007 from people in Monsanto, in which they create a transgenic corn expressing double-strand RNA for Western corn rootwort. So, and here is the effect of this transgenic plant. You can see in the right, a transgenic corn is protected from the damage. On the other hand, in the left, you have the non-transgenic crops with a significant reduction in the amount of roots. So probably very soon, the IRAC will have to include one new group, because RNAi it's not included yet in this table. So here I will describe some in brief some experimental design I mean, suppose you have a very important pests like stink bugs that are very important in many parts of the world, including here in Brazil. So these insects, they are sucking insects and they feed on pods. And here you have the damage they cause. So they are very important. And what is needed for RNAi? I mean, you need genetic sequences. Without genetic sequences, you can do anything. You cannot do anything with RNAi because you need genetic sequences. And how you do this? Well, you have to extract eggs, larvae, pup, adults, do a pool, and then you sequence this. That's why it's very important to make this available that other people, they can access this and also work with RNAi. So once you do this, you have to spend some time and do all the bioinformatic work. I will not go in details here, but normally in my lab, we do some processing, assembly, and do the annotation. I mean, there are a lot of different protocols today, very well established, that people can use for processing, assembly, and do annotation of the genes. Once you have the genes, like in this example here, for Euschistus zeros, which is one stink bug, very important soybean in Brazil, we identify all genes related to RNAi machine learning. Once you have this, you have to synthesize double-strand RNA, and double-strand RNA that you target your genes of interest. Normally, they're critical genes for development. Well, here in my lab, as a start point, we use a double-strand RNA with 400 base pairs. From 200 to 800, it's a good start. Also, it's important to design this double-strand RNA with one eye in between conserved and non-conserved regions. If you select your double-strand RNA in conserved regions, probably will narrow the spectrum. But if you design in non-conserved region, probably you will broaden the spectrum. Important always to do some search to see target closely related species, and of course off-target. Off-target is something that I don't care too much, because off-target, in my opinion, is not too important, but of course non-target effect, they are very important. Once you design your double-strand RNA, you have to synthesize, and then you can do this in vitro, like commercial kits. You can do some constructs in E. coli. I mean, there are a lot of different protocols. They are not difficult. And also you can buy commercially. Here I have some images of my previous PhD student of double-strand RNA delivery. You can do by irrigation water. I mean, you just put in the water, and then plant can absorb double-strand RNA together with the water, and then when insect feed on the plant, I mean, they uptake double-strand RNA. Also, you can do by soaking. I mean, we put the seedling in an Eppendorf tube with double-strand RNA. We let 24 hours and then put insect to feed on this. And also, in the case for a stink bugs, they are very greedy insects. I mean, they feed in almost anything you put in front of them. So we let them for starvation for a couple of hours, and then we put a drop of water, of course, it's a solution of double-strand RNA, and then the insect, they uptake the double-strand RNA. Now I would like to describe some of the results of one of my previous PhD student, Daisy. Well, Daisy explored two, should I say, still new technologies like RNAi and CRISPR in neotropical stink bug Euschistus zeros. So, in fact, her thesis was composed for 6 chapters. I will try to describe here 3 chapters, which initially we generated data set. As I told before, we need genetic sequence. And for this insect, we had no information in the public banks like GeneBank. And then we use the RNAi. So here, he initially we generate our data set, and we identify 47 genes that are involved in RNAi mechanism in different compartments, but I'll not talk about this. And then when we prove that RNAi related genes, they are present in the transcriptome of our target insect, well, and then you can say, yes, RNAi may work for this species. In other hand, indicates the functionality of the gene silencing machinery. And then we run out 3 experiments: double-strand RNA stability, gene silencing assay, and then, of course, we also access the expression of RNAi core genes. And then here you can see in the left image, we just put double-strand RNA from GFP. You can see over the time there is no degradation. But here in the right, we mix hemolymph from our insect and double-strand RNA of GFP. And you can see here, after 10 minutes of incubation, you see some degradation, half an hour more degradation, 1 hour, and after 2 hours, all double-strand RNA was gone. And this, it's something that you need to think, because enzymes that are in hemolymph or even in cell leaf, they can degrade this RNA. And then we conduct more experiments. Here we inject double-strand RNA of VATPase, which is one gene very used for pest control. And you can see here in the left that over the time, the insects, was increasing the mortality over the time. You can see 96 hours, about 35 of insects, they die. And of course, here you can see in 48, 72, and 96 hours, you can see here one down regulation of double-strand RNA of VATPase, which means that RNAi it's working. And also we investigate the expression of 2 very important genes that they are part of RNAi core machinery. And you can see here in the left, DICER2, there is one up regulation, you see, in 24, 48, 72, and 96 up regulation. And then here also one up regulation of Argonaute 2. What that means? It means that when double-strand RNA is injected, and then you see one upregulation of these genes, it means that RNAi machinery is responsive to this double-strand RNA, which is injected and enter in the system. So we'll conclude here that we found 47 genes related to RNAi. And yes, Schistosauros has proven to be sensitive to RNAi upon double-strand RNA injection. And of course, as I told before, it's very important to make this transcriptome public, and that people, they can access and also they can do their research. Well, one second article we published here, we used the parental RNAi, which means that we inject double-strand RNA in the females, and then you see the effect in the little babies, I mean in progeny. So here we evaluate 5 candidate genes like labial, deformant, and the proboscidea, and the potential of these genes be involved in the rostrum formation. I mean, if you silence this gene and then the labium is deformed and the insect cannot feed, so that was our motivation for this experiment. So, as I told, when you use parental RNAi, we inject in females and then we observe in the next generation. So you select some females and then we inject here double-strand RNA, 10 microgram per insect, and then we let a couple of hours to insects take a brief, and then we put some males, and then the females they formate, and you collect the eggs for 20 days. Okay, and then we access daily fecundity, and I mean number of eggs, hatching rates, and gene size, of course. So here you see the results. I mean, here you have in the left, you have here the number of fertilized eggs per female per day. It's number of eggs, okay? And you see here that this DFD was the only gene that presented here one difference from others. And here in the right, you have the hatching rates, 71 for GFP, which is totally expected, and then less hatching rates for other gene sizes. I mean, 4%, 12%, 6%, 1%, and 66%. But we are interested here in abnormal phenotypes because we were searching for genes that potentially could cause deformation in the rostrum. So we collected this insect with abnormal phenotype, and here we have one image. What is one normal phenotype? You can see here the labium. This is how it looks like, one normal insect. And then we have here some abnormal phenotypes. For this DFD gene, you have here two indicated here by this red arrow, two short and curly structures close to the rostrum, you see here. And then for SCR and Proboscipedia, we had here like a bifurcation or like so bifurcation in the rostrum. And this last image here, I'm not sure, maybe you cannot see, but there is some claws here. So it's a rostrum with some parts of legs. So interesting that all these abnormal phenotypes, they're unable to feed and they die. Okay. So in this experiment, we proved that the 5 genes tested were involved in Schistosauros embryonic development. The knockdown of some genes lead to abnormal rostrum development. And of course, the parental RNAi has shown one efficient to study function of genes in Schistosauros. Well, if you reach this point in which you generate your data set, your genetic synthesis, you do some experiments in the lab that you prove that your insect is sensitive to RNAi. You prove that when you inject or you supply your insect with double-strand RNA, the RNAi machinery responds. For this, it's time to think on how you deliver this strategy to the field. So, and then it is time to think on your insect. It's a suck insect, it's a shell insect, or is a root feed. This is very important because delivery is one of the most important steps in RNAi. So you have basically 2 different strategies for delivery RNAi in the field. You have the transformative RNAi that can be from transgenic plants or transplantomics plants. As I told you, I mean, we have been using BT crops for a couple of decades. They are very efficient. They work quite well. They provide a long-term control, but of course they have one extensive regulatory process. Sometimes they have a high cost, and sometimes they're not applied to every crop and pest. Like for perennials, many fruits, sometimes you don't have straightforward protocols for plant transformation, and sometimes it takes years for plant transformation. So it's not applied. On the other hand, you have non-transformative RNAi that you can do synthesis of double-strand RNA. You can spray, inject, or drench, and also you can do some strategy. I will not talk too much on VIGs, but I have one PhD student who is working with VIGs. And when you do non-transformative RNAi, of course, is GMO-free, is suitable for any crop and pest in thesis. And of course, when the problem is when the plants grow, new leaves arrive, and then you need to spray again. You need a continuous supply of double-strand RNA. Well, supposing here you have a plant cell, and you have some plant pathogens. You have here some insects that are sucking insect, like stink bugs, and you have here some cherry insect, like grasshoppers. And then you spray here double-strand RNA on plant leaf. And well, there are two main pathways that this double-strand RNA can enter into the cell of these organisms. I mean, like number one is it's very obvious, like the cherry insects, they will uptake double-strand RNA directly by feeding. But in the case of stink bugs, it's not so obvious. And for fungi, we know that these organisms, they can take up double-strand RNA directly when you spray on leaves. But then the second pathway, we know that the double-strand RNA can enter into the plant cell, and then, of course, into the phloem cells. Insects like stink bugs, they can take up, and of course, grasshoppers and cherry insects, and of course, fungi as well. And then, of course, this double-strand RNA will process by dicer-like from plants, and then with very short double-strand RNA, fungi can take up as well. We know that insects, they need double-strand RNA large molecules. I mean, like 300 base pairs, 400 base pairs. And we know that very small double-strand RNA, they are not able to activate the RNAi in insects. But in case of fungi, we know already that even, large double-strand RNA and small double-strand RNA can activate the mechanism in fungi. So when you talk about the spray of double-strand RNA, we need to think in two main things. First, cost-effective methods for production of RNA. RNA is a very delicate molecule. I mean, it's difficult to make in large volumes, and pure RNA is very difficult. So, and second is stability and delivery. I mean, RNA, it's a very delicate molecule and can degrade in leaf surface and also in soil and also in insect gut, because there are a lot of enzymes. As I told you, a lot of enzymes in cell leaf as well can degrade very quickly RNA. So here, I just would like to show you how important it is to produce pure RNA. I mean, you can see here, this image shows the survival when you use non-purified RNA. You see here, in 72 hours and 96 hours, a lot of organisms, they survive. But when you use purified RNA, no organism was alive. It means that RNA should be very pure, otherwise you will not have efficient control strategy. So when you're talking mass production and formulation of double-strand RNA, you can use a method called chemical, which is very expensive and is limited for short RNAs. You can use a method called in vitro transcription, which you can scale up for kilos, but is costly method. You can use fermentation, which is easy, cheap. You can scale up for tons, but it's a dirty RNA. And then, of course, you have to clean this, and to clean, you spend money. There is one company which I have been in contact a couple of years. It's formerly EPSI, now it's RNA Agri, which they developed one very clever strategy for produce high amounts of RNA at a cost of $2 per gram. And just to our knowledge, it's when you deliver this to the field, this is proposed to be 1 to 10 grams per hectare. So in this strategy from EPSI, they use some microbes, and then they do some co-transfection of 2 plasmid. One plasmid has the RNAi construct here in red, and another construct with the coat protein. When they are co-transfected into the microbe, and this region here and this region here, they are complementary. And then when both are expressed, they self-coupling. And then by the end of the process, you have the RNA, and surrounding the RNA, you have the coat protein. And then, of course, you do some cellular lysis, in which we take out the bacteria, and then you have a particle purification. With this, you can just spray out in the field. You can break down this cover protein, and then you can do new formulations. Another very smart strategy is designed by NanoSur, which is a small company, in which they modify the RNA. This structure they call modified double-strand RNA, they call MDS, in which they modify the structure of RNA. And of course, with this modification, the enzymes cannot degrade this RNA. And they believe that this will, of course, improve the translocation across cellular membranes, and of course, prevent the rapid degradation of RNAi. And by the end of process, of course, increasing the efficacy of RNA-based pesticides. On safety for humans and mammals, I mean, on the last decades, a lot of research in medicine demonstrated that it's very difficult to get RNA into the human mammal cells. I mean, when you eat a lot of RNA every day, but this RNA does not go into your cell. So and also we have a lot of enzymes in organs and blood, intestine, stomach, and kidney and mouth that they degrade the RNA. Also in the soil, you can see here that about 99% of the double-strand RNA, it's gone in between 24 to 48 hours. So, and the remaining double-strand RNA, which is about 1%, that persists in soil and plant tissue. So also, life of RNA is not easy. So in the soil also there is a very fast degradation. Well, I can say that RNA, it's a natural process and it's GMO free. RNA can act in past insects, and of course, as it's a technology that is tailor-made. I mean, you can customize to be very active toward the pest insects, but without harm beneficials. And you can use, as I told, to make insects susceptible again in case of resistance, the same as for weeds. And the good thing is that the big companies like Monsanto, Syngenta, they are making their own process to produce double-strand RNA, which will make the price goes down. I mean, 10 years ago, the prices were very high to small amounts of double-strand RNA. And also, it's important to mention that many small, should I say small companies compared to others like RNA Agri and Ansur, they are doing their own process to producing double-strand RNA, which will push down the prices. Just to have an idea, in 2006, you pay about 12,000 to make 1 gram of double-strand RNA. In 2016, like AgroRNA, which is a Chinese company, was selling 100 grams of double-strand RNA for $4,500, which means $45 per 1 gram of double-strand RNA. So with more people, with more small companies developing their own methods to produce double-strand RNA, probably the price of 1 gram will be lower than now. So, well, I think that RNA in agriculture, which can be used for pest insect, for control plant pathogens. One thing you have to think is it's in between efficiency and cost, efficiency and cost. And how is the efficiency of this strategy and how is the cost of this strategy? We know that RNAi is not working very efficiently for many species. And I'm not sure if you make this available for many pest insects and plant pathogens, because it does not work very well in all species. So this is something you have to discuss about efficiency and how will be the costs to deliver this strategy to the field. And it's very important, it's essential to define the delivery strategy. If you use transgenic plants, well, as I told you, they are very efficient, they work quite well, but sometimes not applied to every crop. And also, there are other strategies like topical application or VIGs or maybe Symbions also. So it's something we still have to discuss in the coming years. So this is just one picture of my students in my lab. And I think that what I had prepared for today is this. Thank you.
Moises, I'd like to thank you very much for your presentation. So following the schedule, I would like to invite Dr. Rene Hunter to present his talk. So when you can share your screen now, please. Okay, Wayne, you can start now.
you hear me, Eduardo?
Yes, I'm hearing
Okay, I had the mute on. I just took a while. It took a while to figure it out. That was just an excellent overall coverage of RNAi. That was just done very, very well. And for people who are not in the field, I hope they really paid attention on Dr. Zoe's presentation, because that really covered sort of where we're at right now in RNAi. I know it changes in different countries as to whether it's a GMO or not a GMO. So what I've been doing is trying to optimize the double-stranded RNA trigger to make it more effective, which means we can put less on a crop plant, and so we can reduce the cost overall. And so we've got it down to our estimates on these companies that can mass produce are around $45 to $50 an acre. You have to convert that to hectare, sorry. But it's comparable to some of the miticide insecticides where citrus growers are paying $45 an acre. And so the biopesticide technology is coming way down and getting there. But like the previous speaker said, in China, they do bacterial production, which is a very dirty prep, and then they just irrigate it across the field. But it does work. And I think as more people start using it like this, we'll see the safety issues start to fall away. I'm a research scientist, primarily insect vectors, hemipteran insect vectors of plant disease. I'm at the Fort Pierce lab in Florida, which is on the eastern coast of the United States. We're north of Brazil, and I was lucky enough to have Dr. Eduardo Andrade come to my lab, and we were able to collaborate on an RNAi project that covered about 4 years and turned out to be really successful. So I'm going to talk about some of the things I have done in collaboration and some of the things we are currently doing to optimize the RNAi trigger. So the primary pests that I work on are these three hemipterans, the psyllid, the whitefly, and the glassy-winged sharpshooter, or leafhoppers in general. They all transmit a variety of pathogens from bacteria to viruses. They all occur globally, and they're important economic agricultural pests. When Eduardo was here, he came in I think it was 2011, stayed to 2014. In our collaboration, we were able to file and submit and get awarded the first RNAi patent on this Asian citrus psyllid, covering the territory of the United States. And we produced several feeding bioassay papers where Eduardo was able, you saw some pictures in the previous presentation where we can use whole plants with soil applied double-stranded RNA in water, or we can use cuttings, whether it's a leaf or new growth cutting or a flower. We can absorb the double-stranded RNA quite readily into plant parts, including the roots. So my focus has been primarily using genomics background approach to identify the genes and interactions that are essential for the psyllid to either acquire and transmit, or to develop, or to molt. Like was said previously, we don't always have to kill the insect. We have products that can just reduce the fecundity, because we have paid millions of dollars to import and establish parasitoids of these insects. And so we don't want to just remove the food of those beneficial insects, but with RNAi, we can reduce the pest insect without harming the parasitoid. And so the other problem was we have to get our product inside the plant. So you can see the picture there to the right, which is a scanning electron micrograph produced with microtomography. This is work I did with Alvaro Tercidor, who's a professor at University Granada in Spain. And we were able to capture a psyllid as it probed into the vascular tissue. You can see the stylet bundle. That's the head of the insect there with the eyes on the left and the right, compound eyes that are all bumpy. And then the labium coming down and the stylet bundle into the vascular tissue of the leaf, of the rib of the leaf. And so Eduardo and I worked a lot on getting delivery to make sure we could get the concentration we needed. And a big part of the thing with hemipteran insects is you have to get your double-stranded RNA trigger into the vascular tissue. In leafhoppers, you want it primarily in the xylem, but in the psyllid, we want it primarily in the phloem. And our rough calculations is that we could get an effect with about 2 picograms of double-strand RNA in the vascular tissue. After 6 days of feeding, that would induce the RNAi response in the psyllids. So different insects would ingest more. Leafhoppers ingest constantly all the time. They would get a higher dose. Whiteflies and psyllids, their females are often disrupted by males wanting to mate. And so there's a lot more movement and activity. But at night, they settle down and feed. And so this was sort of an important thing, that we could get a lethal dose inside the plant. Now, we were able to validate this using this fluorescent labeled double-strand RNA kit, which is there at the bottom of the screen, the Ulysses nucleic acid labeling kit. It's a very good kit. Labeling the guanine does not disrupt the activity of the trigger. It does not disrupt your capacity to do PCR. And you can visualize it inside the plant and insect tissue. So it was a very good way. We were able to detect down to 0.004 nanograms of double-strand RNA in a citrus leaf. So it's a very good kit. I recommend it for anyone working in RNAi. Just a basic background on the experiments. I'm going to cover some experiments in brief, but how our double-strand RNA is synthesized. We have moved from unmodified. In my lab, I work primarily now with modified double-strand RNA, but the unmodified double-strand RNA, you heard, can be made in many different ways. Megascript kit, or we're always using agro-RNA, which is now, I think, called Genolutions out of South Korea. It's been very reliable. Now there's Greenlight Biosciences and several other companies that can produce kilogram quantities. The modified kit uses a modification at the 2' position, similar to the NanoSur company. They have a different modification attached at that same location. But this kit, and there's like 4 patents on it, not by me, but by the guy who invented the kit, attaches a fluorine attached cytosine or a fluorine uracil at that 2' location. And this reduces the RNase A capacity to degrade the double-strand RNA. And so we get stability even, we've done trials in animal hemolymph and blood sera, and it's amazing how much we can increase the survival of our molecule and it remains active. And so I'll be showing some data on that today. My controls are either the Chinese sac brood capsid double-strand RNA, which is a non-insect homologous sequence from the work I did with biologics on the bRNAi stuff years ago, or it's a green fluorescent protein, which works very well also. Our concentration of triggers in whole plants can range anywhere from 10 to 100 milligrams, while leaf cuttings in smaller tissue amounts will range from 10 nanograms to 1 milligram. And all these references, I can email them, or Eduardo can email them to anyone that wants them. Just send me a reply by the email. So the first trial I'm showing here is we did an RNA design. And when we originally started, we always would design 3 double-strand RNAs. Now I deal with long double-strand RNA in the sense that it's normally between 200 and 360 nucleotides. We find the best results around 240, 250 nucleotides. You can make shorter ones. The shortest length they recommend is not to go under 70 nucleotides, and then you can make concatenamers of different triggers, 70, 70, 70. But even then, I extend to 90 if I make a concatenamer of 2 or more triggers. And In this case, I wanted to know which of the double-stranded RNA regions was the best to use. Now, this is unmodified, just using our standard plant absorption feeding assay. And you can see that the difference in mortality between the different regions, 95%, 82%, 59%, while the treated GFP was just 6.2%. And so if I wanted to select one of these to patent or develop further, I'd really am going to focus on the one that's 95.5%. I probably would include the 82, but the 95 one is the one I really would put a lot of time and money towards validating. And this is just showing that visually, the differences. And trehalase is a very important enzyme in insects. It's a separate clade, so it separates out. It doesn't it separates out of all the other trehalases like in plants. And so there's no problem with putting it in plants. It doesn't match anything. I then took that same, just the one that was the best, and we ran a trial against insecticidal soap. Most people have in their lab safer soap. And we did a trial against 5% safer soap. Now this is topically applied. The previous one was soil applied and the insects fed on whole plants. This is topically sprayed onto the leaves. The double-stranded RNA is just in water and it absorbs into the cuttings. And you can see we get a pretty good mortality even as compared to a safer soap. The insecticidal soap problem was that it dries and then it's not active. When the insects just walk over top of it, it wasn't causing any mortality at that point. We only sprayed the plants once, and in nature, out in the field, in Florida anyway, we get fog every morning. And so those plants, if there's a soluble insecticide on the leaf, it rehydrates and would be active again. But in the lab, you know, it's not using the atmospheric moisture, it's just in the lab. And the double-stranded RNA outperformed the 5% soap. And so that was most of the work when Eduardo and I are working together. And I started working in 2014 with a company that works with antisense oligonucleotides. And I started another project with morpholinos that was also a modified backbone structure. And when you start reading the literature on the modified nucleotides for human medicine and drug development, much like we have for COVID, you can find that these things are extremely stable and it increases their activity because the modified nucleotides can have a higher affinity to binding to their complement. So G to C pairing can be much stronger, more specific. And so in this trial, I used a modified double-stranded RNA, both the same sequence of arginine kinase in the leafhopper, glassine sharpshooter leafhopper, but one has modifications in it. And you can, so the BBs in the middle there are significantly different from the controls, but your mortality is still at only around 20%. And I use arginine kinase as my positive double-stranded RNA control because it gives me activity between 20 and 40% pretty consistently. And then the modified, it jumps. And so you see a jump of mortality. The highest jump there was 30%. So this theoretically, when you're developing, I talked to a lot of people on developing a biopesticide. If you have something that's only active at 70%, but for whatever reason in your system, it's the trigger and target that you want, you can use a modified and get a bump closer to 90 to 100% from that same molecule by just using and incorporating these modified nucleotides. And so this was, I think, where everyone's going to go. This is what we're seeing with the NanoSir company. This is Greenlight Biosciences. Even though they mass produce unmodified, they will have to eventually move to developing and mass producing modified double-stranded RNA. It gives the product more stability, more consistency. And until that happens, commercial companies aren't going to risk spending hundreds of millions of dollars to commercialize it. So to give some examples of using these modified pyrimidines, this paper just got accepted a couple of days ago. And what it is I grouped all the data I had where we used modified pyrimidines and we fed them through plant tissue to whiteflies, psyllids, or leafhoppers. Now this shows that if you incorporate somewhere between 30 and 55% of your nucleotide in your double-stranded RNA, this is C's and U's. We tried samples where we replaced all the nucleotides and it got worse. And then when we replaced just the G's, you end up with it forming G blocks. And so it tangles up. And so it just becomes unusable. And so the C and the U are were the best modifications, replacements in the double-stranded RNA that we can find. And that's also what the patents are on. So I imagine they had already screened that as well, but we did it to prove it to ourselves. But we can get this increase in mortality. The lower box there is just explaining that a lot of these methods have been published. And so you can see some references there, and they can be found online, or we can send them to anyone who wants them. This is an example of what that would look like. So this is syntaxinate 1A, double-stranded RNA, and this is the sequence we used in Bemisia tabaci, the whitefly. The red letters there are the modifieds that are replaced, substitutions. This is using the modifieds when we fed to psyllids on citrus cuttings. So we absorb the double-stranded RNA, it's just in water, we absorb it into citrus cuttings. This is cactin, which is related to the cuticle pathway, and very important, all the growers want us to get rid of the adults. They don't want to see any adults. So for years we were trying to affect the adults, and now what we're doing is targeting nymph development. And this works really well at increasing mortality across nymph developments. You can see that the two center columns that are CC, one is a modified cactin at 1 microgram. And these are total concentrations, not per microliter. That's total concentration absorbed into the plant tissue. And the unmodified 2 micrograms. And it took 2 micrograms of the unmodified to equal 1 microgram of the modified when you're talking about psyllid mortality. And when you use 2 micrograms of the modified cactin there, the bar on the far right, you can see you get almost 60% mortality. So you're getting a jump almost 20% there by just incorporating the modified nucleotides. And whiteflies on syntaxin, which I showed you the structure prior. Here our control is green fluorescent protein double-stranded RNA, 40 micrograms in an 8-day feeding period on okra seedlings. So these, we root cuttings of okra, and then we use, the plants are about 5 centimeters in height, so they're really small. And again, we were able to get 79%, almost 80% significant mortality compared to the unmodified syntaxin at 66%. And then the controls down there at 17%. And so this trend holds up every time we run these trials. This is the relative expression or suppression of expression level in the whitefly. And again, you can see that the modified syntaxin double-stranded RNA is significantly different from the unmodified. In leafhopper, we get a similar result. We ended up with the modified cactin and the unmodified at the same concentrations, again on treated okra, a 10-day feeding period. And this was for the nymphs, even though the picture is a picture of an adult. And we get higher nymph mortality when they're feeding on a modified version of the double-stranded RNA trigger than the unmodified. So that's where the program we're developing now has gone. The other part of the program is I deal with these these are single-stranded modified nucleotides. These pheno oligonucleotides and also the morpholinos are single-stranded, but they have modifications, more than one type of modification. It's not just the 2'F location. They also have modifications of the backbone and sugars. These traditionally have been called gapmers because it's like nucleotide sequence, then RNA sequence in the middle. So the enzyme will work and the RISC complex will grab it. And then again, the DNA sequence. But by doing this, 4 nucleotides on either end, they make it highly specific, so it will only bind to the complement sequence. And so it greatly reduces any off target. And this is a medical company that's working on this, AUM Life Tech. They also have an agriculture division recently that's branching out. They've got patents pending in malaria, HIV, and a couple other technologies. You go to their website, you can see this technology in more depth. But the main thing was that it's stable and it auto delivers. I don't need to bind anything to this. Just get it inside whatever I want and it will be stable enough and it will auto deliver into the cells. And so this was a great advantage, especially when we can't target Liberobacter or any of the bacteria or phytoplasmas with the RNAi. We were wanting something that would work for citrus greening or Wonglong bean, the worst bacterial pathogen in citrus. And so FANAs fit the bill perfectly, and we were able to suppress Liberobacter, reduce it in citrus as well as in potato and tomato. And so we were issued a patent on that. The top one is the Morpholino stuff, targeting the Liberobacter. And the second one is pending for the FANA, which is targeting not only the insect, but the insect endosymbionts. We can downregulate and target those, which then cause mortality of the psyllid, leafhopper, or whitefly. And these molecules have been working really well. And these are just some recent pubs that came out in the last couple of years. One of them again came out like last month. So this is all brand new work that we're doing. You know, we've been working on it for 5 years, but we're publishing it now. And I think it will be well received. I hope that people will try it and see if it can solve some of the problems that they're facing. So here is targeting the Liberobacter in the psyllid vector. So as I said, we can target the pathogen in the psyllid vector because it replicates, but we can also target the endosymbionts in the psyllid. The main thing with this FANA technology is it's not as easy to design as was shown by Dr. Zodi's work. You know, RNAi is really straightforward. I don't think most people have trouble designing things. You still have to think about what region and what target you want, because it has to be expressed. You don't want to target the formation of sperm and feed it to the nymphs. That's not going to work. You have to target the life stage that's producing your target if you want to suppress it. It's the same with the FANAs. Also, with the FANAs, because they're only 21 to 22 nucleotides long, we make an average of 8 products to target 1 mRNA. And they do have a software that can help you predict, but they're not as cheap as double-stranded RNA, so it's a little more expensive, and you can't make them yourself, at least with the FANA. I have made 2OMEs, just order them online, and you can make those and screen those. This is a totally different patent than the 2OMEs or the 2MOEs. So, but this worked really well. We had 3 of the 4 designs, the ones that are labeled BBB, significantly reduced the Liberobacter and psyllids that were infected with Liberobacter. And you can see product number 3 there just didn't do anything at all. It just did not work. And this is typical. You'll get usually 2 out of 4 that will work. This is a soil applied treatment. We did soil in citrus, tomatoes, and potatoes. And in citrus, this is targeting the gyrase. All these bacteria depend on gyrase for replication. And it was shown by the Nobel laureate at Yale University, Sid Altman, who I collaborated with on the Morpholinos. They target a very narrow 13 to 14 nucleotides with the Morpholinos. But with the FANAs, I wanted more length for specificity to make it more specific to the region I'm trying to target in the endosymbionts. So we use 21 to 22 nucleotide links with the FANA products. I hope that's clear, but you can see from this that we targeted the endosymbiont, Prophtella armatura. I should have put the genus there, sorry, Prophtella armatura. Prophtella is inversely associated with the Liberobacter and the Psyllid. So when it is overabundant, the Liberobacter is suppressed naturally. The thing is, in the system, Liberobacter in the Psyllid, combined with the presence of Wolbachia, almost completely suppresses Prophtella armatura. And it's necessary to do that in order to acquire and transmit the pathogen. But in this study, we're able to show if we totally suppress Prophtera, one of the important Psyllid endosymbionts, just for normal nutrition and feeding, that we got 80% mortality in the insect. So whatever the Prophtella is providing, it's not providing enough to the Psyllid. So one of the things we also did was we did CRISPR, the first CRISPR targeting to produce mutants. You heard in the previous talk also talking about CRISPR. We are now on the verge of people having edible CRISPR or transient CRISPR delivery through plasmids or symbionts that can be used to the work we did was to show that we can inject adult females and produce second generation mutants with the thioredoxin knockout. And this produced adults that had a greatly reduced lifespan. It was only 6 to 8 days, while the wild type adults would have an adult lifespan between 18 and 22 days at the temperature that this study was conducted. It decreased eggs tremendously. Those adults that only lived 6 to 8 days, over the entire 8 days, only produced 3 or 4 eggs. Well, the wild type were sitting over there producing, these were the mock treated, they were injected with just the water, 156 eggs in their lifetime, but they had a much longer lifetime. So this is one strategy we're also looking at for population suppression or modification. The new trend now is to not use the word control anymore when we're talking about insect management. They want to go to a softer technology where we don't have to kill things, but we can just reduce their fitness enough that nature can sort of suppress the insects. And then Eduardo sent me this slide. I wanted to acknowledge the people that contributed to the seminar as well as to some of the studies. My program has greatly benefited by being associated with people from Brazil and Embrapa. Before this program, I didn't know you guys existed. I'm embarrassed about that. And I am just so impressed with how far Brazil science is ahead of us. And it's good that they're ahead because it gives us something to try to collaborate with and to work towards. And I just really appreciate all of the work and collaboration that Eduardo and I have had. And that's it. Thank you.
Okay, Rene, thank you for your presentation. You bring some new information, I think, for all the viewers. So now you can unshare your screen so we can all have our pictures here so we can start our discussion. So we have some questions came from YouTube. So I make the question for you, okay? For you both. So the first, Moisés, você está aí, Moisés?
Yes.
Okay, so the first question is for you, Moisés.
Okay.
From YouTube. Américo Viana made the question. The high nuclease activity of stick bugs is a limitation for the use of RNAi as a biopesticide. How to overcome or reduce these limitations?
Okay, so I think it's not a limitation, but it's something that we need to deal with. We know that insect they want to protect themselves from the new strategies, and I think that the enzymes that they're produced in the saliva or even in the stomach and the gut of insects, they can degrade all double-stranded RNA. That's why you need, otherwise you need a continued amount of double-stranded RNA, or continued supply of double-stranded RNA. But I think that it is a long discussion because this will depend very much on how will be the delivery strategy and how is the insect itself. In the case of stink bugs, I think that it's not a problem to not use RNAi. There are a lot of strategies you can do to protect the double-strand RNA, and these all they are optimized in the lab before we deliver the technology in the field. But I don't think that enzyme or activity that it's degrade double-strand RNA is something that you need. Okay, now there is some degradation, then you cannot use this. It's something that you can deal and you can overcome this. There are a lot of strategies to protect double-strand RNA, and but this, if you are talking on spray out in the leaves, like I demonstrated that we know already that double-strand RNA can go into the plant cell, into the phloem cell, and then the stink bugs, they can uptake double-strand RNA. But then you have to deal to protect the double-strand RNA. I think that the best answer I can provide now is this. But of course, you can open and you can stay all afternoon discussing on this.
Okay, just catching this topic. We know that stink bugs, they usually like to feed on pods and seeds on the plant. There's a specific tissue, so how can you deliver to these tissues? I guess is another issue to control stink bugs.
Yeah, I don't know. I mean, I think it is very difficult because specifically in that, maybe you can, because when you do some transgenic plants, you can organize that the double-strand RNA will be expressed in specific regions. But of course, I mean, I need more information to answer this. You know that some transgenic protocols, you can generate plants that you produce the double-strand RNA, the things you are planning to express in the plant, that you express in a specific parts of plant, like only in the leaves, only in the roots. But I don't know exactly, Eduardo. But yes, it's possible, but we need to get more information.
Okay. Wayne, talk about this problem with nuclease activity. You already have some information or results of using this antisense or modified double-strand RNA to be working on recalcitrant insects.
Okay, you want me to, the question is to address the increase in activity?
My question is, do you have any results of using this modified double-strand RNA to work on some kind of insect is recalcitrant or they have high nuclease activity on the gut?
The modified double-strand RNAs work really well outdoors, indoors, in the lab, outside. They can be absorbed in plants without adding anything to them. I was able to show we can we've got this clay delivery system, which we call slow delivery, that's being, the company is now working with, what is it? Greenlight Biosciences. The company is Oil Dry that makes a clay pellet. I could mix that, double-strand RNA absorbed to the clay, mix it into the soil, and every time the plant is watered, the double-strand RNA, it leaches out, and you get double-strand RNA delivery for over a year. Unprotected double-strand RNA over a year. The protected double-strand RNA also delivers. There's no difference in delivery in the concentrations. And I think this is also going to be a very big way to not do GMOs where people just want to protect a temporary protection for part of the year, like in a row crop vegetable, right? So I don't know if I'm addressing your question exactly. It would help me if you type the question in the chat. I could see more specifically.
Okay. So we have another question for you, Wayne. Maria Laura Garcia asks you if you wonder about amount of double-strand RNA you need to get to the insect to have your the RNAi effect.
Right. So in the hemipterans that you saw, the whitefly, the psyllid, and the leafhopper, if you look at most studies, there's only like, I think, 1 or 2 that report activity before 3 days, which we've never been able to see. Normally, when you feed these insects RNAi or double-strand RNA products, you don't see any expression suppression till day 4 or 5, and you see mortality, if that's what you're measuring, on day 6 through day whatever you're measuring, day 15. The concentration we've been able to calculate down to 2 picograms for the whitefly and psyllid in the plant tissue. If you can get 2 picograms, which doesn't sound like much, but we're treating a 1 gram piece of new growth cutting or a small ochre plant that's about 1 to 2 grams in weight with 10 nanograms of double-strand RNA. Because these insects are ingesting, we did a waste, like a honeydew trial, a excreta trial, and the psyllid is excreting roughly 2 microliters a day during feeding. And so we calculated at 2 picograms, and we don't see an effect till like day 6. It's going to take 4 or 5 days for the insect to reach that concentration that will trigger the RNAi strong enough to cause the effect. Now with the modifieds, that's probably going to be shortened by 1 or 2 days. But it does, like Dr. Zoe said, it does not take a lot of double-strand RNA if you have the right trigger.
Okay. We have a question from Moises. Moises, about non-transformative RNAi. Even using a non-conservative sequence, a wide range of double-strand RNA products can likely will be less harmful to the environment than chemical insecticides. So is the RNAi insecticides of the future?
Well, yeah. Also, it's a very complex question. Well, the first thing, I mean, there is no, in terms of specificity and selectivity, there is no parallel in the market. I mean, you can compare regular pesticides, insecticides with RNAi. It's I mean, it's completely different. I mean, but it depends on how you design your double-strand RNA. And I mean, we do not have conducted experiments to prove this, but my experience said that even if you design a double-strand RNA in a non-conserved region, even though it will be more specific to the insect comparing to other pesticides, because it's something that you need to plan before. I mean, the selection of your target, which you can find a specific target for your insect, or at least that is only in the insect and not in mammals. It's only for this fact, it's more specific and more selective to other pesticides. And the question is, I think that even though if you select in non-conserved, of course, you broaden the spectrum. Otherwise, you will be more narrow on the spectrum. But even though it's far more selective and restricted to your organism. I think that if I could not answer properly, please let me know, and then you can find another way. But I think that is, in general, is this.
Oh, so there another question on the opposite side. They ask if you choose a very conservative region, could be more safe, but there's a chance that a resistant insect could rise by mutations, or it's very difficult to have this kind of situation?
Well, as I told, RNAi, in my opinion, it's less prone for resistance development, but that is based in the current resistance mechanism, like the mutations, because we know that the RNAi can be activated even if some mismatch occur between double-strand RNA you synthesize. So, and normally we see like the mutations, they occur in one base pair. And then for heme pair, the RNAi activation, it should be many mutations one by one. And this is, as long as you know, it's almost impossible to occur. So, but of course, as I say again, based in current resistant mechanisms, I think that will be difficult. But of course, if you do not use RNAi technology properly, insects, they find a way. Soon, if you don't use it properly, probably you find the insect resistance will arise.
Okay. Wayne, we had a question for you. So the antisense technology looks like a good alternative to insecticide. How it will be the most important limitations for field application, like temperature, sunlight, water quality? How do you expect to be the most limitations for this technology to use in the field?
So you're asking what would be the greatest limitation to implementing RNAi biopesticides?
Yes, more specific to this new antisense technology, the modified RNA, double-strand RNA. Did you understand?
The microphone is muted.
Wayne?
Yep, you're right. Microphone was muted. Okay, so for aptamers and oligos that are being developed with the modified nucleotides for livestock and plants and human medicine, in agriculture, the last 2 years have seen major reduction in production costs because agriculture is going to benefit because the medical profession in drug development has dramatically increased and built factories. Hundreds of millions of dollars have been poured in the last 5 years to build multiple factories that will mass produce modified nucleotides for the drug industry. And what this has done is drop the price for those of us who wish to use that technology in agriculture. So
You're muted.
today is less than $8.
You say $8 per gram or per milligram?
Okay, so that was 10, back then, that would have been like 100 nanograms was $8 in 2011 to treat beehives. But for treating a field, on my calculations for citrus, I need 200 milligrams to treat 100 trees. So you're looking at like half a gram per acre, because not all of it's going to work. Well, it'll work, but it's not all going to get absorbed into the plant. So if you overcalculate even 1 gram per acre, if your target only needs nanogram amounts, that becomes feasible. If you need microgram amounts, now you're talking 10 grams per acre. And if you need, larger concentrations to have an effect, then you're talking, kilogram quantities. But even a kilogram is supposedly now getting down to under $100 if you have a contract, you're going to mass produce and spray 500 acres or 1,000 hectares, right? It's a production cost. The more you get, the cheaper it is.
Okay, so do you think that this new modified technology for double-strand RNA could reduce the amount of double-strand RNA needed to apply in the field, as you've shown with your results?
Right, that's the whole focus, is to try to find the lowest possible active dose at the lowest possible cost to the grower, right?
And these modified RNAs, they can reduce the cost to reducing the amount needed for application in the field.
Correct.
But how about the price of this modified double-strand RNA to mass produce?
Mass production is becoming affordable. It's already affordable. NanoSur does not produce for other people. He's a company, but he produces it in a way that's profitable for him to mass produce, and he's hoping to get a commercial agreement to sell it. Greenlight Biosciences also says they can produce a kilogram for $2, but they don't tell you the percent activity. And that's the key point, is what is your percent activity? Is that 100%? Because no one produces 100% active AI, active ingredient. So instead of 1 kilogram, you'd have to order 1.5 kilograms.
And you're talking about normal double-strand RNA, but
yes,
about this modified double-strand RNA. So the cost is expensive.
If unmodified double-strand RNA is sold at $45, again, for 200 trees in citrus, that's doable. The modified ones have an added cost, and you're running between $75 and $80. But another couple years, that should be as cheap as $50 an acre. The price has dropped every year for producing it.
So in your vision, and this question is for you and for Moises. So how long do you think you have the first commercial product available for growers in the United States, like a topical RNA product?
Right. Well, in the United States, Monsanto pulled their product when they got bought by Bayer Crop Science. And Syngenta China was the last company that had a submission for, I think, potato beetle topical application. And the last time I talked to the person at the Environmental Protection Agency, EPA, in the United States, they said all submissions had been pulled.
Do you know
So
any,
no one. So Bayer is probably the most likely to resubmit something in the coming year. Bayer Crop Science.
and how EPA is treating the RNA-based products, like a regular insecticide or a biological insecticide, how the vision of EPA?
How is it going to be registered? Is that the
Yes,
question?
or regulated
It will
by
be,
EPA.
it will be registered under biopesticide. And currently that means you have to meet the requirements of like a GMO plant. They're using those steps so they don't have to develop new steps. They're trying to just use the requirements for a GMO plant to satisfy the requirements of an RNAi biopesticide. So some of those steps don't apply, right? Because you don't make protein, so you don't have to do an antigenicity test. But that's just the framework they're currently trying to use.
Okay. So, Moises, how do you envision?
Yeah. Well, I'm not sure exactly, but in Brazil, you don't have available in the market for agriculture any RNAi-based product. And but since 2018, they already approved the regulation, and RNAi-based products, even though when you spray on leaves or to transgenic plants, will not be considered as a GMO. The RNAi biopesticides, they will be considered from the English the precision breeding innovation, like the PPI. So they will not be considered one GMO strategy for agriculture. That's all I know. But I have no idea if some of these big companies already apply for registration in Brazil, any product based on RNAi. At the moment, is all I know.
Okay. There's another question for you, Moises. When you talk about non-conservative RNAi, you say that probably we need to think in constant supply of RNA, double strand RNA. So you constantly spray as new growth appears in the plant. But do you think that the systemic movement of the double strand RNA molecule inside the plant can help to reduce the need for constant spraying?
Well, I mean, that's well, as you know that if you create a transgenic plant, you have a continuous supply of double-strand RNA. Every time that insect feed on plant, they will uptake double-strand RNA. But when you use one spray, and of course, this double-strand RNA will remain for a certain period, and then as the plant grow, new leaves will come out, and then you have to spray again. And of course, you know that the RNAi using this strategy is always transient, and then the insect, they will uptake the double-strand RNAi mechanism will be activated, and then for a certain period and then it's gone. So probably if you spray, you have to continue to spray. I mean, I'm not sure, I mean, how long this can remain in the field, because we are working on the lab. Maybe Dr. Wayne can explain better, because I know that Wayne has a lot of trials in the field, and then this can, I mean, address better this question.
Right. So this is one of the big, the big debates now. It's a very good question. And this is how it's different from chemicals, hard chemicals, right? There's chemicals that can last for years in the field. And there's ones that can last for months. And then there's ones that degrade in weeks. And double-stranded RNA is a biopesticide, by definition, I guess, has to degrade at least within months. It cannot persist for a real long time. And if you want it to persist, and Dr. Zoe's right, you have to put it in a GMO plant or some kind of expression system that will be there and constantly express it. The great advantage of having a short survival in the field is all your vegetable and row crops, whose growing cycle is 60 days or less. You don't need a long-term chemical. And this is where RNAi is going to really have the advantage, because it's safe to eat for humans.
So, Rene, did you comment that in Canada they approved RNAi product in 2019? Can you give us more information?
Yeah, I was trying to find the announcement. At the 2018 meeting, we had the International Congress of Entomology, Bill Moore, Dr. Moore and I. Moore used to work for Monsanto. He's now the RNAi lead researcher for Bear Crop Science. We invited the equivalent of the Environmental Protection Agency representative from Canada Ag Science, I don't remember their title. And she said they had already approved double-stranded RNA as a treatment for protecting vegetable crops. Primarily, they were targeting flea beetles. And those are patents held by Wired, Dr. Wired, who's shown also specificity of double-stranded RNA. So Canada is ahead of us, ahead of the United States anyway, in approving these.
And they were the first to approve the transgenic maize too, for cotton root worm. So I have a question now about your latest developments on targeting endosymbionts. So do you think that could be a new strategy or new tool to control insects using targeting the endosymbionts?
Yes, you saw the, I don't know if I don't have the reference right here, but I can send it to you. There's a brand new paper by Nancy Moran's group, who's the world expert in endosymbionts. They transformed the honeybee endosymbiont to express double-stranded RNA. And so this is coming, this is going to be a common thing in another 5 years, using the endosymbionts to express. And this is why you don't want to kill the insect. You want to modulate it, because if you kill it, you get rid of your endosymbiont, right? But if you put it in there and you just reduce fecundity or make it no longer able to acquire and transmit the pathogen, the focus now is to convert vectors into non-vectors.
It's a clever strategy. Well, I'm very curious about the CRISPR technology. How do you deliver the CRISPR technology to the insects? The same as double-stranded RNA?
You're saying how long to transfer the technology?
No, how do you deliver the CRISPR technology that is shown to the insect?
Oh, you can microinject insects. That's not a problem. Like I said, there's 2 papers now, mine and another one by a group in California that does gene drives, and you can inject adult insect, females, that will cause mutations in the embryos inside the female. So then you can select for a population that has the modification. So honeybees are like cattle. In the United States, they're considered livestock, and you can manipulate them and have queens that are modified, and all the offspring will be modified. The question I think you're asking is, how would I do that for a psyllid or a leafhopper when it's a
Yes.
wild population? And this is where you're talking about gene drive. Now, gene drive system still scares the public, and it's having a really hard time to open the door. But the technology is maturing very, very fast. And there's several companies now developing gene drive for very difficult to control vectors of human pathogens. And that's where the door is going to open. With the pandemic, people understand you have to control the pathogen or the thing that transmits the pathogen. And that's going to help all of us.
Wow. Yeah. It's an interesting future. I'm curious about, you talk about the pharma, and you said something that you need to design more than one molecule to target the gene. So when you deliver the pharma molecule, you need to deliver more than one sequence to have a good effect.
Is that to me or?
Yes, that's for you.
Yeah, so when you were in country with me and we were working, we read papers that said a concatemer of 2 targets has no effect. The most recent papers now say concatemers have an effect, and we've already shown in my lab also, we can make concatemers. I made a design of 3 different targets into 1 double-stranded RNA, and we got really good knockdown of the insect.
Wow. Yeah, I'm trying
So
to.
it depends on your selection, selecting the right target.
Uh-huh. Okay. So we have no more questions on YouTube and my list. So I'm here to thank for you both to have your time to give these wonderful talks on this webinar hosted by PROCISUR. And in the name of the organizing committee, I'd like to thank you very much for your presentations. Thank you very much.
Thank you, Eduardo.
Thank you, Eduardo.
Yes, so now we'll finish up your presentations for today. Thank you very much for our attendees. And this presentation will be recorded and will be available on the PROCISUR website and YouTube.
Okay, bye. Bye, everyone.
Goodbye.